Hello, my name is Irina Shakarova, and I'll be discussing the semester-long experiment that we had with fava bean plants, aphids, and lady beetles. I'm going to start off by discussing the background information first. The fava bean plants produce sap, which is what aphids are going after for a source of food. Aphids are herbivore insects that reproduce sexually at a rapid pace and feed on the sap of plants. Lady beetles are predators insects that feed on aphids and other insects. They are commonly used to eradicate an aphid outbreak because the average lady beetle can consume 50 to 60 aphids a day. In this experiment, there will be 3 plants. My hypothesis is if aphids are added to one plant, then the plant will have a slow increase in height and mass. If both aphids and lady beetles are added to another plant, then the plant will have a faster increase in height and mass than the first plant. However, if nothing is added to the last plant, then the plant will have the fastest increase in height and mass. The method of this experiment began in the first week of the class when we have planted the beans into the pot, watered them, and left them in the classroom underneath the lamps. The way we distinguished our plants was labeling the pots with table number 5 and the 4 symbols. As it says right here, that's how they looked. One was labeled as a star, one was labeled with 2 lines, one was labeled with a circle, and one was a triangle. Later on, the triangle pot represented the control group. The star pot represented the aphids, and the 2-line pot was aphids and lady beetles. All of the measurements were obtained by an inch ruler. The first measurement of the plants was calculated 2 weeks after planting the source. Then it was measured every week. After the plant reached the height of 20 centimeters, we have gently removed the smallest plant from the triangle pot, avoiding ripping off the roots, and then washed off as much of the soil as possible to measure the length and estimating dry mass of the plant after gently tapping it dry. Then we proceeded by cutting a decent piece of size of aluminum foil and placed it onto the scale to measure how much the weight of the foil was before adding the plant and wrapping it into it to proceed by measuring it again. The mass measurement resulted in being 4.4790 grams. The wrapped plant was then placed into the drying machine and left there until the last week of class, leaving the rest of the plants seedling to expand. Later on in this experiment, we have built the greenhouse cages using an empty plastic soda bottle, rubber bands, and mesh. The mesh was to provide a natural environment for the bugs. We have used the 3 remaining plant pots to study the change of length and mass due to lady beetles and aphids being added. One of the plants was not tampered with to act as a control group, and the other with the aphids, and the last was both aphids and lady beetles. The bugs were placed into the cages by our lab TAs. The only down factor of the study was there wasn't enough aphids and lady beetles placed into each plant because of the size of the classrooms. So therefore, we only received 1 to 2 bugs in our cages. In order to prevent the escape of the bugs, we taped the mesh from the bottom of the pot to the top and placed a sheet of mesh onto the top, keeping it closed with a tight rubber band. Each week we took off the bottom mesh with caution and measured the length of each plant. The results were calculated into a lab notebook, and this is the general diagram of how it looked whenever we had the pods in our cages. Okay, so the conclusion of this experiment is that our hypothesis was partially correct. The control group plants did not instantly decrease in length because no predators were involved in their trial, but in our hypothesis, we predicted that it would have the fastest increase, which resulted in being false. It had the slowest increase. The plants with aphids had an increase in length, but then suddenly decreased after the bugs were added, which confirmed that our hypothesis was correct. The plants with the aphids and lady beetles also had the highest increase in length, but then decreased a little more than the plant with just the aphids after the bugs were added, which concluded that our hypothesis was again correct. The final mass of the triangle plant was taken out on the 4th week and placed into the drying machine, which resulted in being 5.87 grams. The final mass for that plant in the control group was 0.955 grams. And the final measurement was 10.5 inches. The final mass for the plant with the aphids, the star pot, was 0.557 grams, and the final measurement was 10.5 inches. The final mass for the plant with the aphids and lady beetles, which was the 2-line pot, was 0.7036 grams, and the final measurement was 13 inches. In conclusion, the mass was the highest for the control group. The length of the control and the aphids plant was the same, but the mass for the aphid plants was the lowest. The length of the aphids and lady beetles is the highest, but the mass calculation is the median of the other two plots. According to the PowerPoint shared by RTA of the plant growth class data, my experimental design results were very similar to the rest of the class. The graphs show that the plant with just the aphids had a decrease, but the plants with the aphids and lady beetles had a slightly higher decreasing rate of the plants. Now that we have come to the end of our video, I would like to give out some credits to the people that made this all happen. Jake helped me work on my script, and Luis did a fantastic job with the data table and the graphs. Last but not least, a big thank you to Taylor, our cameraman.